Hello, everyone. I'm Dr. Carmen Pugliafito, and I'm here today with Dr. Andrew Moshfeghi, who's a professor at the USC Roski Eye Institute. Welcome to Retina Synthesis once again, Andrew.
Thanks. Pleasure to be here.
So today we're going to talk about the Ocular Therapeutics tyrosinase kinase inhibitor and the hydrogel delivery system. Can you tell us a little bit about the rationale for using tyrosinase kinase inhibitors, or TKIs, in the treatment of wet macular degeneration?
Sure. I think a lot of us retina specialists understand the unmet need that is durability of the treatment effect, the good treatment effect that we've come to expect with anti-VEGF monotherapy, if only it would last much longer than 1 to 3 months that we've become accustomed to. So we're looking for new therapies that may have novel mechanisms of action that may allow it to have a prolonged duration of action. And so tyrosine kinase inhibitors have the advantage of, number one, having been on the market for other indications for quite some time now. Axitinib in particular was approved for renal cell carcinoma, and it's a small molecule, so it can penetrate the retina nicely, get to the site of action, and then after being conjugated with this proprietary hydrogel platform, can then stick around for quite a long time and continue to work on the choroidal neovascular membrane.
So we know that the core of the currently approved treatments for wet macular degeneration are inhibition of VEGF by antibodies or placental growth factor blockers. How do TKIs work against the VEGF pathway?
Sure. So, they work by a similar mechanism of action. This particular one is a highly selective inhibitor of the VEGF receptors 1, 2, and 3, as well as platelet-derived growth factor receptor as well. And so they then work intracellularly, given their very small molecule status, to interfere with cellular signaling with respect to the actions of these receptor tyrosine kinases, and by that way end up effectively being VEGF inhibitors, as well as having some effect on PDGF as well. Are you frozen?
So tell us a little bit about the hydrogel delivery system. I'm loud and clear.
Oh, yeah, no, you were frozen on my end for about 20 seconds. So the hydrogel delivery system, which is used in a variety of products with this study sponsor, consists of a polyethylene glycol-based hydrogel platform. And basically, this is customized with the drug. So it integrates with the drug during the manufacturing process, such that it is planned to completely biodegrade via ester hydrolysis over time. And it's biocompatible, so it doesn't leave like a wrapper or remnant behind. There's no cast that's left behind as it biodegrades. And it really has low potential for inflammation because of these characteristics. That's engineered to deliver the drug to dissolve slowly over time in either as little as days in some applications or as long as several months with this drug. We anticipate with axitinib conjugation that the goal would be to deliver axitinib steadily at near zero order kinetics for between 6 to 9 months.
So you gave us an update at the angiogenesis meeting about the phase 1 trial of this molecule. Can you summarize the results of the trial?
Sure. So this is interim analysis of the study that was carried out in Australia. It's a phase 1 first in human study. And this was a dose escalation study consisting of 4 cohorts. And we have data to report on 3 of them so far. And basically, this was a low dose 200 microgram group, a medium dose 400 microgram cohort, and then cohort 3A consisted of a 600 microgram group, as well as cohort 3B, which was the medium dose plus a baseline anti-VEGF agent. Now, keep in mind in this study, patients in this study were the most advanced types of recalcitrant wet AMD patients in cohorts 1 and 2. And then as dose escalation occurred, investigators started enrolling more typical patients later on in the study. And so then the study endpoint was at month 6 with follow-up thereafter, and patients could be retreated according to retreatment criteria as well as investigator discretion. And what we had was that a relatively well-balanced group of patients at baseline. These are patients with pretty low levels of visual acuity, as well as very thick retinas. There was no patient in this study who had baseline central retinal subfield thickness of less than 400 microns. And cohort 1 had nearly 700 microns of average baseline central retinal thickness. And then what we did is we looked at patients based upon, different cohorts, as well as whether they were previously treated or treatment naive, because both types of patients were allowed in this wet AMD study. And we have data which suggests that there is a dose escalation effect such that the low dose group, we did not see a consistent treatment effect, whereas with 400 and 600 microgram groups, we saw more robust central retinal thickness effect on OCT, as well as visual acuity effect as measured by ETDRS visual acuity. And not surprisingly, the medium and higher dose groups had the most robust benefit, and we did not observe signs of inflammation. We had a nice durability. After all, the first question you asked me was, why even look at a drug like this? The whole reason to do that is to try to get a similar effect to what we've already seen with anti-VEGF drugs for a longer duration of action. And what we've seen is that over 60% of all subjects treated had a durable treatment effect of 6 months or longer. And even 50% of patients had durability of 7.5 months or longer, based on the data to date in this open label study. We ended up sharing several subjects from cohorts 3 as well as cohorts 4, which were the medium and actually I misspoke there. We ended up showing several subjects from the study who had nice detergescence of the macula over the first several months, with a prolonged treatment effect out to month 6 and beyond for both cohorts 3 as well as cohort 2.
Yeah, there were a number of treatment naive patients that had impressive results over a year, really.
Yeah, with cohort 3, we found that almost 80% of the patients were treatment naive. And we think that this was due to the investigators kind of becoming comfortable with enrolling patients in this study after the initial cohorts had been filled. And they did quite well. But this is relatively small number so far. We have to eventually study this in a larger numbers of patients to see if that same treatment effect bears out.
So there are plans underway based on this, plans underway for a phase 1B trial. Can you tell us about that, how it's designed?
Sure. So there's a phase 1B study already underway, this one in the United States. It's prospective, multi-center, double-masked parallel group study. that has patients with neovascular AMD, which was controlled with existing usual care anti-VEGF monotherapy. And the goal is, unlike the first study in Australia, where patients were coming in with active exudation that we were then trying to make go away with OTX TKI, in this US phase 1b study, these patients are coming in with controlled exudation. So that, yes, they had demonstrable prior exudation that got under control with anti-VEGF, and now we're trying to maintain that fluid-free macula by exposing them to OTX TKI at baseline, as well as a mandated baseline anti-VEGF injection.
When did they get the anti-VEGF injection? At 1 month?
They get the anti-VEGF injection at 1 month, which is a little bit different than other studies where patients might get both treatments at baseline. But in this study, we're trying to take advantage of the previous usual care therapy during the run-in period that kept that patient dry to begin with, but then also expose them to another anti-VEGF, so that over the first couple of months, the patient has a fluid-free macula while the TKI starts its intracellular activity and kind of starts working and kicking in roughly around the month 2 time point. So then afterwards, we're relying on the OTX TKI by itself in the active treatment arm to sort of do the work of keeping the macula dry. There's also a control arm with aflibercept Q2 months as a active control with a sham OTX TKI injection at baseline.
Well, I think your treatment approach makes a lot of sense because really what you want the TKI to do is to keep the retina dry and vision good. And you can count on traditional anti-VEGFs to stabilize the situation initially.
Yeah, the one thing that people don't necessarily recognize with TKIs is that, if you're using them as a monotherapy, they don't kick in and start working right away, like in the first 1 to 7 days after exposure, like an anti-VEGF agent does. And so you kind of need something that's around first to kind of get things under control. So that's where these run-in injections, as well as the mandated injection at month 1, comes into play.
So in the phase 1A trial, the patients were only given 1 implant. Is that correct?
Well, they were only given 1 episode of exposure to OTX TKI. However, with that first generation injector, the maximum solitary dose that could be achieved was 200 micrograms. So, which is fine if you're only shooting for 200 micrograms, but if you want 400 or 600, you had to give 2 or 3 injections in the same episode. With this new second generation injector that's employed in the US study, a solitary 600 microgram injector is utilized.
But they didn't, but what I...
Yeah, there was no subsequent re-exposure to OTX TKI in the Australian study. That's correct. Yeah.
So I think the results are, the durability results are impressive. It's small numbers, of course.
Yeah.
And a heterogeneous group of patients, but very interesting. And I think TKIs have a role in the future as a durability agent, maybe not as solo agents, but in combination with traditional anti-VEGF therapy.
Yeah, we are excited to see this data and look forward to seeing what additional studies show us.
Okay, Andrew, thanks a lot for your time, and we'll stay tuned about OTX TKI.
Thank you.